Hello everyone, welcome to BioBoost. Today we will cover eukaryotic replication, which is almost same as of prokaryotic except the initiation and termination processes. Looking at the cell cycle again, here you can see in eukaryotes also S phase is the synthesis phase where DNA gets replicated and histone gets synthesized. So here we will see how eukaryotic replication is different than prokaryotes one. So here also replication is semi-conservative, it is semi-continuous, but the DNA is linear here and the chromosomes are linear. So they have multiple origins of replication because in one go you can't replicate the whole DNA. So it is comparatively slower than prokaryotes due to complex chromatin structure. And as the DNA is linear, we will see how their forks meet, how the termination takes place, and how the ends of lagging strands get replicated. Looking at the origin of replication, here you can see the chromosome. In prokaryotes, it is circular, while in eukaryotes, it is linear, so multiple origins are there. And there are ARS, which is autonomously replicating sequences, which helps in autonomous replication of DNA without any host. In comparative study, you can see in prokaryotes, there is replication fork that is single and it is bubble-like, and end of replication two daughter cells are there. In eukaryotes it has complex chromatin structure, so at the end you can see on the linear bubbles are formed like this, and multiple origins are there for multiple purpose, and at the end the strands get replicated here you can see. Now the change in enzymes of DNA polymerases and proteins which are involved in DNA replication of eukaryotes and prokaryotes are it is a very important table where you can see the function of each and every enzyme. ORIC is there for origin, MCM is the eukaryotic helicase here, and CDC6 which is basically cell division cycle, which is 6. So it loads helicase, while pol alpha and primase are there for synthesis of primer. Fen 1 is the pol 1 here, pol delta and pol epsilon is pol 3 here in eukaryotes. RPA are there, which are the SSB proteins. DNA ligase 1, topoisomerase second is there as gyrase, and proliferating cell nuclear antigen PCNA are the sliding camp here, and replication factors RPC are loader of sliding camp in eukaryotes. So the first process is initiation and replication in this diagram. It is very important to know at what phase of cycle these are loaded. In G1 phase, origin of replication is found. It is the beginning of replication which initiate at G1 phase only. Here MCM2 helicase is incoming in G1 phase. So with that loaders of helicase, loaders of this MCM helicase comes CDC6 and CD2, CDT1. Then in G1 to S transition, pre replication complex is formed and the MCM helicase unwinds the DNA. Now in S phase, the helicase are loaded. So this is very important question from exam point of view. Like at what phase of cycle helicases are loaded? So S phase is the phase. Here sliding clamps are there. These are also loaded, and initiation occurs only once per cycle to prevent re-replication. So this is very important point in eukaryotes. Replicosomes are formed at initiation phase till now. So step 1 is done of initiation. Next is elongation, where polymerases are needed for extension. So polymerase alpha synthesize RNA DNA primer using primase activity, while polymerase delta elongates the lagging strand. Pol epsilon elongates the leading strand, and Pol gamma replicates mitochondrial DNA. Role of primase in eukaryotes is that it is a part of DNA polymerase alpha complex. It synthesizes a short primer for elongation. Now, if we see the process, then it occurs in 5 to 3 direction continuously, and Pol epsilon is responsible for it. 5 to 3 strand here you can see in diagram. Here Pol epsilon is responsible to synthesize. And on lagging strand, Pol delta is responsible for making Okazaki fragments or discontinuously synthesize DNA strand. Okazaki fragments are shorter in eukaryotes compared to prokaryotes. Here you can see SSB proteins are needed to stabilize the unstranded means single stranded DNA because it is unwinded now and it needs stability. So Pol alpha is the first responsible to make the first stretch of DNA after primer addition. So Pol alpha works at lagging strand and it makes the first strand of DNA after primer addition, while Pol delta is responsible for extension on leading strand. And primer is added here. You can see ligase is responsible to join the next. Now, Okazaki fragments in eukaryotes, it is 100 to 150 nucleotides long and it requires an RNA primer. Now, RNA primer are joined, but they are removed after the fragments are joined by RNase H enzyme. DNA polymerase delta then fills the nick, which is left by the RNA primer removal by forming phosphodiester bonds. So you can see DNA poly delta extended. Now the medium means the middle part is replicated normally, but the ends of DNA you can see on lagging strand there is addition of primer because you can't stretch it in 3 to 5 directions. So on lagging strand the end gets unreplicated here. So after each replication the chromosome will get shortened. This end is called telomere. We know the tail of chromosome. It gets depleted if not replicated in next generation. So it will cause several diseases, this shortening of chromosomes, and it might lead to cancer like diseases. So, for this, an enzyme is responsible to replicate this portion, which is called as telomerase. So, you need to remember that telomerase works on the lagging strand where the last fragment stays unreplicated because you can't add primer to that. So what happens? Telomerase is a ribonucleic protein enzyme, means it's also ribo origin. It's also a RNA template. So it contains an RNA template and it extends the three end of the telomeres. It is active in germ cells, stem cells, and cancer cells. So this enzyme prevents loss of genetic material during replication. Here you can see on the parent strand, you can see at the three end it is unreplicated on the lagging strand. So telomerase, the green enzyme, comes with an RNA template on it which is complementary to the parent strand. Here telomere is synthesized. The RNA template is added to it and the enzyme responsible to now polymerize it, catalyze the strand is DNA pol alpha. You know, on lagging strand, DNA pol alpha is responsible. So on lagging strand, DNA pol alpha comes here to stretch the remaining rest strand. Now, proofreading is also important in eukaryotes. So here it is performed by DNA pols, which possess 3 to 5 exonuclease proofreading activity, and mismatch repair mechanisms take place to increase the accuracy, which we will cover in next videos. And it is very essential for stability. Now, the last process, which is the termination of eukaryotic replication, it is very important. You can see when bubbles are made, multiple origins are there. So at some end, the forks will meet. Here you can see the left and right fork are meeting and helicases are still loaded here. So what happens here, you can see both the strands coming at an end. Here termination will take place. The leading strand is there and Okazaki on the lagging strand are also made. MCM helicase is there which is loaded. So what happens to unload it? A ubiquitination process is required, which ubiquinate, which destroys this enzyme. Here SF3 ubiquitin ligase complex comes, a protein in which MCM gets ubiquitinated. But the most interesting part is that it is not ubiquitinated here. It gets temporarily unloaded. So what happens? A different protein is responsible to stop its degradation and only disassembly of MCM helicase is takes place, in which the active MCM helicase get disassembled and remains inactive till the next cycle of replication is initiated. So thank you so much.